Welcome to UGC series of lectures. This time I will discuss the fruit development and its maturation. Let us first discuss fruit development. Botanically speaking, fruit consists of seeds enclosed in a mature ovary of a flowering plant. The wall of the ovary grows along with seeds, and as they develop, the ovary wall dries out to become a pod, as in legumes, or becomes a capsule, as in poppy. In fleshy fruits, a fruit is the fleshy part of a plant that surrounds the seeds. In morphological terms, fruit is the structure that arises from the ovary or several ovaries, and in some cases associated floral pods after fertilization. Fruit is an evolutionary adaptation unique to flowering plants and is designed to aid in the dispersal of seeds by various agencies including animals, wind, and water. Since fruits develop from organs of the flower and thus involve differentiation or redifferentiation of pre-existing organs, evolutionarily floral organs represent modified leaves, and so the fruit is also a modified leaf. Fruits serve two functions. Number one, to protect the seeds during development, and number two, to disperse the seeds following maturation. Although most fruits develop from the ovary, and in fact fruits that consist of ripened ovaries alone, such as the tomato and pea pod, are called true fruits, while fruits that consist of ripened ovaries and other pods, as in the apple, are called accessory fruits or false fruits. In true fruits, the outside of the fruit is called the pericarp and develops from the ovary wall. The pericarp can be dry and papery, as in maple, or it can be woody, as in nuts, or even it can be fleshy, as in berries and stone fruits. The pericarp of some fruits is further differentiated into specialized layers called exocarp, that is fruit sac, mesocarp, that is fruit flesh, and endocarp, that is inner cell layers. An exocarp will develop a cuticle and may display a variety of morphological features such as coarse hairs, as in kiwi fruit, or fine hairs, as in peach. Exocarp plus cuticle restrict gas exchange and determine general appearance of ripening fruit. Mesocarp tissues usually represent the fleshy part of a fruit and commonly hold chloroplast and starch grains. In fleshy fruits such as berries like tomato, kiwi fruit, and grapes, this tissue typically comprises large parenchyma cells and contains the main vascular network. Endocarps are less common but typically develops a dense hard case around a seed, as in peach and apricot. Now, phases of fruit development. Physiologically and biochemically, fruit development can be divided into four phases, that are fruit set, phase of cell division, phase of cell elongation, and number fourth, that is fruit ripening. Fruit set. This phase includes ovary development in the flower, and following anthesis, a decision to abort or proceed with further development. Fruit set is normally dependent on pollination. Fruit growth is provoked by this pollination, followed by pollen tube growth, and fertilization. Pollen triggers fruit development, indicating that positive signals are generated during pollination. In the absence of these signals, the flowers abscise. Growing pollen produces gibberellic acid, and lagging slightly behind the growing pollen tube is a wave of increased auxin production by the style and then the ovary. Continued fruit development usually relies on the continued presence of developing seeds. Seed abortion or removal causes fruit abortion, which can be reversed with auxin application. Phase of cell division. This phase involves a period of rapid cell division. It involves all growing parts of the fruit. This is thought to be controlled by the developing seeds. The number of fertilized ovules in a fruit is correlated with both the initial cell division rate and the final size of the fruit. In most fruits, cell division occurs in the first several weeks after flowering and represents the first 20 to 30% of the fruit's growing season. Cell division occurs in many cells in the fruit simultaneously, and as the number of cells increase, there are more cells to divide. Consequently, the number of cells and the fruit weight increases at a faster rate in the early season. This is called the exponential phase of growth. There is a correlation between cytokinin levels in the developing embryos and cell division in surrounding tissues. But there is no direct evidence that embryo cytokinin is in fact regulating fruit cell division. Phase of cell elongation. This phase is the period of most rapid growth, when cell division more or less ceases and growth is almost exclusively by cell enlargement. Final fruit size is dependent on cell number, that is cell division, and cell size, as well as production of intercellular spaces. In this phase, food reserves are accumulated and most fruits attain their final shape and size before the onset of ripening in the next phase. This phase of fruit growth is mainly accomplished by cell expansion in longitudinal, radial, and tangential planes. Longitudinal growth, where cell enlarge parallel to the long axis of the fruit, will often be a big factor for development of elongate fruit such as cucumber and marrow. Radial growth increases diameter, as in some pumpkins. Cell enlargement is not a uniform process. Cells in various regions of a fruit often enlarge at different rates and in different planes, so that many mature fruits show strong gradients in cell size from their surface to the center. For example, in apple fruit, cells closest to the core are smallest, with cell size increasing towards the fruit surface. Conversely, in many berries such as kiwi fruit and grapes, the smallest cells are found in outer regions of the pericarp, with size increasing progressively towards inner region. Fruit ripening. The last phase, that is fruit ripening, has been well studied because of its importance in fruit harvest and storage. Ripening represents the shift from the protective function to dispersal function of the fruit. Ripening is a biochemical process occurring in fruit between late maturation and early senescence. Ripening transforms a physiologically mature but often inedible plant into a visually attractive fruit with characteristic aromas and flavors. Ripening also makes the fruit more attractive to animals as agents of seed dispersal. Ripening occurs synchronously with seed and embryo maturation. In dry fruits like cereals and nuts, ripening consists of desiccation and is considered maturation. Ripening in fleshy fruits is designed to make the fruit appealing to animals that eat the fruit as a means for seed dispersal. Ripening involves a complex series of events: a change in color, softening of the ovary wall, and changes in sweetness and flavor, which makes fruits both tasty and attractive to eat. Fleshy fruits are either climacteric or non-climacteric. Fruits that show sudden and dramatic rise in biosynthesis of ethylene and consequently higher respiration rate are referred to as climacteric fruits. In others, where there is low level of ethylene production and consequently no or low rise in the respiration rate are referred to as non-climacteric fruits. Ripening in climacteric fruits has been well studied. It is partly because they include many important commercial crops, and partly because in these fruits ethylene acts as an inducer of many genes associated with color change, change in carbohydrate reserves, and softening of the pulp. From transgenic studies of tomato and other plants, it has been concluded that ethylene is a major regulator of the ripening process. There are also developmental factors involved because fruit does not attain competence to respond to ethylene until near the end of the cell expansion phase. Several genes associated with ripening are ethylene inducible. This occurs transcriptionally in most genes, but at least one is known where messenger RNA accumulation is regulated post-transcriptionally. None of these genes are induced until competence for ethylene response is attained. Ethylene production is autocatalytic. That is, exposure to ethylene stimulates the synthesis of more ethylene. This occurs because the genes for the biosynthetic enzymes are ethylene inducible. The result is a positive feedback. Fruit softening involves a partial breakdown of cell wall. Several enzymes are known to be involved in this process. Polygalacturonase hydrolyzes bonds in pectins. The gene for this enzyme is ethylene inducible. Changes in fruit color involves changes in the expression of pigment biosynthetic genes. The major pigment in tomato is a carotenoid. The first committed step in carotenoid biosynthesis is catalyzed by phytoene synthase, and the gene for this enzyme is induced by ethylene. During ripening, fruits show an increase in the concentration of sugars, either by hydrolysis of starch within the fruit or by continued import of sugars from other parts of the plant. Moreover, sucrose is hydrolyzed to hexoses, principally fructose and glucose. Activities of enzymes involved in starch sugar metabolism, such as sucrose phosphate synthase and acid invertase, has been shown to rise in several fruits, and their messenger RNAs have been shown to be upregulated by ethylene. In addition to change in sweetness, flavoring components like citric acid and malic acid increase during ripening and combine to produce the unique flavor and aroma of the ripe fruit. Ripening in non-climacteric fruits. Ripening of non-climacteric fruits has not been studied to the same extent as in climacteric fruits. Although some information is available for some fruits such as strawberry, citrus, and grapes. The ripening of strawberry involves many of the same changes that are seen in the ripening of climacteric fruits, that is, loss of chlorophyll and anthocyanins, accumulation of sucrose, hexoses, and flavoring volatiles like alcohols, aldehydes, and their esters, and softening of pulp as well. Many studies have shown that ripening in strawberry fruit is not affected by exogenous ethylene and ethylene biosynthesis inhibitors or inhibitors of ethylene action do not affect strawberry ripening. Softening of the pulp in the strawberry fruit is accomplished by an array of glucanases and pectinases, similar to those in climacteric fruits, but their induction is not linked to ethylene. Alcohol alkyl transferases are enzymes responsible for the production of esters that contribute to the distinctive tastes and aroma of many fruits. The gene called strawberry alcohol alkyl transferase is expressed much more highly in ripe red fruit than in green or white fruit. In contrast to strawberry, citrus fruit is unusual. Although it is non-climacteric fruit, carotenoid synthesis in the orange peel is ethylene regulated. Fruit maturation. Maturation is the completion of the development of a fruit to the point at which it is physiologically mature enough to be separated from the parent plant. Typically, this is the point at which its seeds are viable. Ripening and maturation can be synonymous for non-climacteric fruits that are edible at the time of picking and have no post-harvest ripening cycle. However, they are quite different for climacteric type of fruits. In climacteric fruits, maturation usually starts before growth stops. Immature plantain and banana fruits are angular, with sharp edges and well-pronounced ridges. As plantain and banana mature, the fruits become more rounded. Further changes include coloration from green to yellow and the development of attractive flavors and aromas. Climacteric fruits such as tomato reach full red color even when harvested green. On the other hand, in non-climacteric fruits such as bell peppers, slight changes in color take place after harvest. Full red color is only obtained while fruit is attached to the plant. As a general rule, the more mature the product, the shorter is its post-harvest life. Importance of fruit development and maturation. The development and maturation of fruit tissues represent a final phase of floral development, typically proceeding and signaled by successful pollination. Although parthenocarpic fruit development occurs, such phenomena typically result from either number 1, genetic alterations that includes gene mutations and changes in genome ploidy, or environmental and or hormonal alterations that ultimately mimic and trigger the fruit development cascade, or number 2, premature embryo abortion that results in fruit with minimal residual seed tissue. Although much is known of the hormonal and physiological signals that trigger fruit development, maturation and ripening are aspects of late floral development for which the molecular regulatory signals remain largely unknown. From the standpoint of agriculture, ripening confers both positive and negative attributes of the resulting commodity. Although ripening imparts desirable flavor, color and texture, considerable expense and crop loss results from negative ripening characteristics. For example, ripening related increase in fruit pathogen susceptibility is a major contributor to fruit loss both before and after harvest. This genetically regulated change in fruit physiology currently necessitates the use of pesticides, post-harvest fumigants and controlled atmosphere storage and shipping mechanism in attempts to minimize loss. In addition to being wasteful of energy and potentially harmful to the environment, such practices represent major expenses in fruit production. Finally, it is important to reiterate that ripening imparts numerous quality and nutritional characteristics upon a significant component of the human diet. Ripening imports various critical aspects of mature fruit, including fiber content and composition, lipid metabolism, and the levels of vitamins and various antioxidants. The ability to understand key control points in global ripening regulation or within specific ripening processes such as carotenoid, flavonoid, vitamin and flavor volatiles will allow for manipulation of nutrition and quality characteristics associated with ripening. The most convincing argument for the promotion of safe plant genetic engineering will be the development of products with direct consumer impact and appeal, such as quality and nutritionally enhanced fruits. I end my today's talk with this note that flowering plants usually require fertilization to form fruit, and fruit can be narrowly defined as mature carpel. The development and maturation of fruits has received considerable scientific scrutiny because of both the uniqueness of such processes to the biology of plants and the importance of fruits as a significant component of the human diet. Thank you.